In this lesson, we are going to learn about how DNA is the code for synthesizing proteins in the ribosomes of all cells. Inside the nucleus of all of our body cells are 46 chromosomes. Chromosomes are made of smaller sections called genes, and genes are made of DNA. Genes and DNA code for the synthesis of proteins. Some important proteins found in the body include all cells are made of protein, enzymes are made of protein, antibodies are made of protein, as well as hormones. So the majority of the living part of you is made out of protein. What you're seeing here is a section of DNA. Those colored bricks represent the 4 bases of DNA, which are adenine, thymine, cytosine, and guanine. Those bases themselves are the actual recipe found within DNA, which then tells our cells which protein molecules to make. The first step of protein synthesis involves an enzyme which will unzip DNA, opening the protein recipe to be copied. DNA is far too valuable to leave the nucleus, could be damaged, so it is opened up so a copy can be made. Messenger RNA, or mRNA for short, is what is going to make the copy of one gene. So what you're looking at there is one gene on a section of DNA that has been opened up. The messenger RNA is making an exact copy of the protein recipe found inside DNA. The major difference between RNA and DNA is that RNA has the letter U for uracil. It does not have a T for thymine. Thymine is only found inside DNA. Uracil is only found inside RNA. Adenine, cytosine, and guanine are still found in mRNA as they are in DNA. The mRNA, which has now made a copy of the protein recipe found in DNA, will now exit the nucleus and head to a ribosome where that information can then be used to construct a protein molecule. Here is a closer look at a molecule of mRNA. mRNA is broken down into sections called codons. Codons are 3 base pairs long, 3 letters long. Each codon codes for one specific amino acid. If you take a bunch of amino acids and link them together, you've created a protein molecule. So in this example, we would have 7 codons, which would mean we would have 7 amino acids, which would mean our protein molecule is made of these 7 smaller building blocks called amino acids. What you're looking at here is the process of translation. The ribosome translates the mRNA into the appropriate amino acids. So it's reading each group of 3 letters, each codon, and then another type of RNA called transfer RNA, or tRNA, is delivering the appropriate amino acid. The tRNA matches up with the mRNA and will deliver the amino acids. You can see them linking together to create our protein molecule. This picture summarizes the entire process of protein synthesis. In the top left corner, you can see the DNA molecule being unzipped by the enzyme. That section, which is called a gene, is then being copied into mRNA through the process of transcription. The mRNA then leaves the nucleus, where it will go to a ribosome. The ribosome then translates the mRNA, reading 3 letters at a time, which is a codon, and the codon will stand for one amino acid. The tRNA delivers the matching amino acids, and when the amino acids have all linked together, the protein molecule has done being created. An easy to understand analogy for the whole process of protein synthesis. You could think of the DNA molecule, or the gene, as the cookbook for making protein molecules within each one of your cells. Messenger RNA is like a copy of the cookbook itself. It's making a copy of the protein recipe found within DNA. Transfer RNA is like the grocery delivery guy. It is delivering all of the ingredients needed to cook up our protein molecule. tRNA is delivering the amino acids needed in order to synthesize the protein. The ribosomes are like the chefs in this equation. They are cooking up the protein itself. They are utilizing the amino acids delivered by the tRNA and following the recipe found in mRNA to synthesize the protein molecule.